Serious ethical issues are being raised after it emerged that US and Chinese scientists have implanted human cells into monkey embryos.
Get your political jokes ready because this story is sure to generate tons of suggestions on how it can be beneficial to everyone in Washington, London, Beijing, Berlin, Pyongyang, and any place else where people seem to be acting like monkeys. A team of neuroscientists has injected electronic instructions into the premotor cortex of monkeys that resulted in the animals getting instructions to complete actions without any other instructions, cues, or stimuli. Can the instruction be to just shut up? What we are showing here is that you don't have to be in a sensory receiving area in order for the subject to have an experience that they can identify. In their study, published in the journal Neuron, neuroscientists Dr. Kevin A. Mazurek and Dr. Mark H. Scheiber describe how they used two rhesus monkeys to demonstrate how instructions can be sent to the premotor cortex using injections of electrical stimulus. The premotor cortex is part of the motor cortex in the brain's frontal lobe that controls the planning, control, and execution of voluntary movements. The premotor cortex feeds directly to the spinal cord, but its functions are not fully understood, which is why these two neurosurgeons met with two rhesus monkeys. The experiment was relatively simple. The monkeys were put in front of a panel of knobs, a great nickname for Congress, and trained to perform one of 4 specific tasks with a knob when one was lit by LEDs. At the same time, a mild microstimulus was applied via implanted electrodes to one of 4 areas in their premotor cortex. This stimulus was just a brief buzz and did not control any of the movements, since the premotor cortex is not part of the brain's perception process. Once the monkeys learned the tasks, the lights were turned off, but the microstimulations continued and the monkeys were able to move the correct nose in the proper way when microbuzzed. To prove that the areas of the premotor cortex were not predisposed to the movements, the researchers switched the electronic impulse injectors around and retrained the subjects. When the lights were turned off, the monkeys continued to move the proper nose. Does this sound like training them to vote? Of course. The researchers say this experiment has nothing to do with mind control in humans. Dr. Mazurek has other plans for it, as he explains in an interview with the New York Times. This could be very important for people who have lost function in areas of their brain due to stroke, injury, or other disease. We can potentially bypass the damaged part of the brain where connections have been lost and deliver information to an intact part of the brain. The example Mazurek gives correlates the experiment to learning that a red light while driving means to put a foot on the brake pedal. If parts of the brain's chain of command to complete this task are damaged, a stimulus could replace them. The next step is to conduct the experiments on humans and eliminate the visual lead stimulus. If that's successful, it means the information or instructions can be injected without the person knowing it. Now that sounds like mind control. Fortunately, before the researchers try this on humans, they will continue to perform their tests on politicians. You knew it was coming.
Serious ethical issues are being raised after it emerged that US and Chinese scientists have implanted human cells into monkey embryos. For practical ethics, I asked her if we should see the embryos as human at all.
I think when we create an organism that is a mix between a human and a monkey, we're really not sure at all what sort of organism we have created. I mean, is it a human? Is it a monkey? Is it something in between? And that raises ethical issues because we don't know how we should treat these creatures ethically. Can we simply use them in scientific experiments? Can we destroy them in experiments when we really don't know what sort of organism they are? About us not being sure what sort of organism we create, how we should treat it, is not so much of a problem for the research we're discussing here, because in that research, as you mentioned, the human-monkey chimeras.
What an ethical mess! According to 2 papers published in National Science Review, scientists in China have successfully cloned monkeys with genetic disease symptoms. Researchers from the Chinese Academy of Sciences used the CRISPR-Cas9 method on in vitro fertilized monkey embryos. With no BMAL1 to help manage their circadian rhythm, the resulting monkeys exhibited a wide range. After taking a fibroblast from the gene-edited monkey, scientists used somatic cell nuclear transfer to replace the nucleus of a monkey egg cell with a fibroblast nucleus. 325 embryos were implanted in 65 surrogate monkeys. All 5 cloned monkeys carried the altered BMAL1 gene introduced in the donor monkey.
For biologists, this term describes any organism whose body consists of cells from 2 different organisms. Why am I telling you about this tonight? Because scientists recently tried creating the world's first human-monkey chimera.
This is exactly what scientists. They integrated human cells into the embryos of a macaque monkey. In simple terms, they created embryos that were part monkey and part human. What happened next? The embryos were grown in the open in a laboratory for 20 days. About it. We still don't fully know what the drawbacks or benefits of creating a human-monkey hybrid could be, but